are the Hot Chillies group. Today we are going to share to you the cell structure which we emphasize on the cellular organelles. We'll start with nucleus. Nucleus is the largest organelle in the cell. It is very important as it controls the cell activities because it contains DNA. The DNA can replicate, which is followed by nuclear division. Nuclear division precedes cell division. Nucleus is enclosed by two membranes which form the nuclear envelope. The outer membrane is continuous with the endoplasmic reticulum. The nuclear envelope is perforated by nuclear pores, which allows the exchange of substances. The nuclear pore has definite formed by the fusion of outer and inner membrane of nuclear envelope. The nucleus contains chromatin. Chromatin composed of coils of DNA bound to a basic protein called histone. Chromatin condenses into more tightly coiled threads called chromosomes during nuclear division. There are two types of chromatins named heterochromatin and euchromatin. Heterochromatin remains tightly coiled during interphase. They form dark patches near nuclear envelope, while euchromatin are loosely coiled during interphase. It contains DNA which is genetically active during interphase. Nucleus also contains nucleolus, which is rounded and darkly stained structure in nucleus. The function of nucleolus is to make ribosomes. It stains intensely because of the large amount of DNA and RNA it contains. Nucleolus is less dense around the central region of the nucleus, where ribosomal RNA is beginning to be folded and combined with protein to make ribosome. That's all about nucleus. Now I'll proceed with lysosome. Lysosome is found in most eukaryotic cells. It is surrounded by single membrane and it is a simple sac containing digestive enzymes. The examples of enzymes present in lysosome are protease, nuclease, and lipase, which helps in hydrolysis reaction. They work best in acid environment, therefore the content of lysosome is acidic. How is lysosome formed? The enzyme of lysosome is made on rough endoplasmic reticulum. While the enzyme is sent to Golgi apparatus to be processed, and the Golgi vesicles containing the processed enzyme will bud off to be lysosomes. Now I'll talk about the functions of lysosome. One of the functions will be the digestion of material taken in by endocytosis. What is endocytosis? Endocytosis is the bulk transport of materials through membrane into the cell. The lysosome fills with vesicle of vacuole formed by endocytosis. Then the enzyme will be released into the vacuole to digest the material inside. The material inside might be taken in for food or defensive purposes. And then the products absorbed and assimilated by cytoplasm, while the vacuole containing undigested remains will be removed through exocytosis, which is the reverse process of endocytosis. The next function will be the turnover of cytoplasmic organelle through autophagy. Autophagy is a process whereby unwanted structures are engulfed and digested within the lysosome. So, the unwanted material will be enclosed by single membrane, which is usually from smooth endoplasmic reticulum. And then the single membrane fuses with lysosome to form autophagic vacuole, where unwanted material is digested. Lysosome also carries out exocytosis, whereby enzymes of lysosome is released from cell. This happens during replacement of cartilage by bone during development. During the remodeling of bone, due to injury or new stresses, the bone might be broken down by lysosome through exocytosis. The sperm also contains ectrosome, which is a type of lysosome, to digest a path through the layers of cells surrounding the egg cell. Lastly, lysosome carries out autolysis, which is self-digestion by releasing the content of lysosome within the cell. This happens a lot during differentiation processes. For example, the tadpole tail is reabsorbed through autolysis. This also happens to the muscles that are not exercised. That's all about lysosome.
Next, we are going to look at ribosomes. Ribosomes are small cytoplasmic granules found in cells. In eukaryotic cells, ribosomes are found either attached to the surface of rough ER or suspended freely in the cytoplasm. Each ribosome consists of two subunits, one large and one small subunit. Each ribosome contains rRNA, which is ribosomal RNA, and proteins in approximately same quantities. There are basically two types of ribosomes, which are the 70S type and the 80S type. S here refer to a unit called Sweatberg unit that is related to the rate of sedimation in centrifuge. 70S type has a diameter of 80 nanometer. It is slightly smaller than the 80S type ribosome. 70S type ribosome is found in prokaryotic cells as well as chloroplasts and mitochondrions in eukaryotic cells. On the other hand, 80S type has a diameter of about 20 nanometer and it is found in eukaryotic cells. As ribosomes are very small, they are the last organelle to be sedimented in a centrifuge. Lastly, about ribosome, the main function of ribosome is acts as the site of protein synthesis. Next, endoplasmic reticulum, ER. ER is an extensive network of flattened sheet-like membrane-bound sacs known as cisternae. They may be covered with ribosomes forming rough ER, or if ribosomes are absent on its surface, it is known as smooth ER. The ER is usually continuous from the outer membrane of nuclear envelope. Next, we are going to look at the functions of ER. Rough ER transport proteins. After the synthesis of a polypeptide chain at the ribosome completes, the protein will be transported by the rough ER and it will be packed in a transport vesicle in order to be transported to the Golgi apparatus for further modification. On the other hand, smooth ER has the function of synthesizing lipids such as phospholipids, which is the main component of the cell surface membrane, and steroid, for example, cholesterol, and sex hormones like testosterone and estrogens. Next, Golgi apparatus. Golgi apparatus is also known as Golgi body or Golgi complex. It consists of a stack of flattened sacs called cisternae. In general, Golgi apparatus acts as the cell's post office. It receives, sorts, and delivers proteins and lipids. More specifically, it adds carbohydrate to proteins, forming glycoproteins. It also produces secretory enzymes, such as those secreted by the pancreas. It transports and modifies lipids too. Besides, Golgi apparatus forms the lysosome. New cell walls in plants are also formed by Golgi vesicles, which contains the cell wall materials. Microvilli. Microvilli are finger-like extensions of the cell surface membrane of some animal cells. Each microvillus contains bundles of actin and myosin filaments, which cause muscle contraction. This allowed the microvilli to remain upright and retain their shape, while still allowing their movement through the interactions of actin and myosin. The function of microvilli is to increase the surface area of the cell surface membrane. For example, it increases the efficiency of absorption of nutrients in small intestine, as microvilli are found in the epithelial cells in the small intestine. Another example is the reabsorption in proximal convoluted tubules of the kidney.
I would like to explain on organelles known as mitochondria. Now, the function of mitochondria is the site of aerobic respiration to produce energy, which is known as ATP. It has various shapes, but commonly known to have a sausage shape. They have double membrane, which is the inner and the outer one. The inner one is a folded version, while the outer one is smooth. Now, the inner version of it is folded to a finger-like structure to increase the total surface area. Now, inside the mitochondria, there are a solution which is known as the matrix. The matrix contains ribosomes, circular DNAs, and also phosphate granules. Now, these finger-like cristae, they project into the solution which is known as the matrix. Now, there are also spaces in between the inner and the outer membrane. The space is called intermembranal space. Now, this site is also for a process known as electron transportation chain. Now, during this process, protons are released into the space, and this causes the pH in the matrix to be lower than the pH in the space. Okay, so now I'll continue on to microtubules. Now, generally, microtubules exist in almost all eukaryotic cells. It made up of a helically arranged globular subunits, which is known as tubulin. Structurally, it's a long, unbranched, hollow, cylindrical organelle. Now, from their walls, they may have cross bridges, like their arms, in order for them to link to adjacent microtubules. Now, microtubules also experience growth, and they experience growth at one end by the addition of tubulin subunits. And this growth is inhibited by colchicine, which is a type of chemical used to investigate the function of microtubule. Now, for the growth process to occur, it requires a template to start, and also a small ring-like structure that have been isolated from the cells, which contains tubular subunits to serve this function. Now, the technique that is used to observe microtubules is called immunofluorescent microscopy. Okay, so now we're going to discuss some centrioles. Centrioles are found in almost all animal cells, and also in lower animal and plant cells, but frequently not in higher level plant cells. So the distinct region where centrioles can be found is known as the centrosome, or either you could call centrosphere. Now, centrioles are made of 9 triplets or microtubules, and at the beginning the centriole replicates and it forms 2 new pairs, and these pairs move to the opposite poles of the spindle fiber, the structure on which the microsomes align. And spindle fibers, they are generally made up of microtubule, and in this process microtubule controls the separation of the chromatid and the chromosome. And as you can see, this is the structure of centrioles. They appear in pairs near the nucleus, and they lie at right angle to each other. And this is a structure where it involves a 9+3 arrangement of the microtubules. Now, basal bodies, they have the similar structure as centriole, and they can be found at the base of cilia and flagella. Now, most probably, based on our research, we found that basal bodies, they are produced due to replication of centrioles. And basal bodies in certain cells, they act as microtubule organizing center, which is known as MTOC, because cilia and flagella, they have a 9+2 microtubule arrangement. So now we're going to see on cilia and flagella. So cilia and flagella, they have the same structure, but flagella is slightly longer than cilia. And what difference is in between them is cilia have a movement where they beat only in one direction, while flagella beats in a wave-like form. Now, both of these are involved in locomotion of cells and also fluids. Now, these beating movements are called by microtubules, and they are responsible for it by underglow sliding motions. Now, generally, in extratip, bacterial flagella have simpler structure with no basal bodies compared to the one you can find in eukaryotic cells. Next, we will learn on cytoskeleton. So, cytoskeleton, they are formed of microtubules in a collective version. And microtubules, they have a long, fairly rigid tube-like structure that acts as a skeleton which contribute to their function as cytoskeleton. Now, plant cells, they have cytoskeleton and can be seen by alignment of microtubules, which corresponds to the alignment of cellulose fibers. Now, indirectly, it helps to determine the shape of the cell, and generally, cytoskeleton can be found under the cell surface membrane.
Chloroplast is a large plastic containing chlorophyll and carrying out photosynthesis. It is surrounded by two membranes and contains a gel-like stroma. A system of membranes are stacked in places to form grana. During photosynthesis, light is absorbed by this grana, which actually consists of stacks of membrane-bound sacs called thylakoids. Thylakoids are the site of light-dependent reaction of photosynthesis, and stroma is a site of light-independent reaction. Grana are connected by intergranular lamella. Grana are interconnected in all directions by intergranular lamella and not just a single string. Chloroplast contains soluble enzymes, starch grains, lipid droplets, circular DNA molecule, and ribosomes. It also contains carotenoid pigments to carrying out photosynthesis. That's all for chloroplast, and now I'm going to talk about vacuole. A vacuole is a fluid-filled sac bounded by a single membrane. Animal cells contain relatively small vacuoles such as phagocytic vacuoles, food vacuoles, autophagic vacuoles, and contractile vacuoles. However, plant cells, notably mature parenchyma cells, have a large central vacuole surrounded by a membrane called the tonoplast. The fluid they contain is called cell sap. It is a concentrated solution of mineral salts, sugars, organic acids, oxygen, carbon dioxide, pigments, and some waste, and also secondary products of metabolism. Vacuoles help to regulate the osmotic properties of cells. Sometimes it contains pigments in solution, including anthocyanins, which color the flowers, fruits, buds, and leaves. They are important in attracting insects, birds for pollination, and seed dispersal. Sometimes plant vacuoles contain hydrolytic enzymes and act as lysosomes.
All the organelles are suspended in the cytoplasm, which is surrounded by a plasma membrane. What makes up a cell membrane actually? Alright, so we have phospholipid, cholesterol, and proteins which make up the cell membrane or plasma membrane. As you can see here, the plasma membrane is made out of phospholipid bilayer. Bilayer means two layers of phospholipid molecules. The thickness of the phospholipid bilayer is about 7 nanometer, and it is mainly made up of protein and lipids. There are some carbohydrate chains above and on the surface of the cell membrane also, such as the glycoprotein and the glycolipid. Glycoprotein is the carbohydrate attached to the protein, while the glycolipid is the carbohydrate attached to the lipid. Okay, now let's zoom into the phospholipid molecule. As you can see here, phospholipid molecule consists of hydrophilic head and hydrophobic tails. Hydrophilic head, they are water-loving, means they will face towards the water, while the tail are hydrophobic tails, they are water-fearing, which they are afraid of water, and they will face inwards, away from the water. They are said to be amphipathic as they have hydrophilic and hydrophobic properties, which enables them to form a bilayer simultaneously. Two phospholipid molecules facing each other, and they will form a layer, and indirectly they will form this hydrophobic interior. Sometimes we can also find some cholesterol in the bilayer. Cholesterol maintains the fluidity of the membrane. At the same time, they increase the flexibility and the stability of the membrane too. As for proteins, they act as receptors and transport substances in and out of the phospholipid bilayer. Plasma membrane is partially permeable, and scientists even named it as fluid mosaic model because it is dynamic. Well, now we are going to the function of plasma membrane. The first function is that it regulates the passage of substances in and out of the cells, and it acts as a boundary between the cytoplasm and the extracellular environment. It also acts as a receptor site in recognizing some external stimuli such as hormones and antigens, and last but not least, it protects the cell from external damage. Now we'll go to the cell wall. Cell wall is a rigid carbohydrate layer of cellulose found outside the plasma membrane. Cellulose has high tensile strength. There are two types of cell wall, which are primary cell wall and secondary cell wall. Primary cell wall are thin and permeable, elastic and strong. Then for secondary cell wall, it is thick and permeable. Some cells do undergo lignification, whereby lignin is deposited in all the cellulose layers. What is the lignin for? It is used to cement the cellulose fibers together. It gives extra strength to the cells, and it even protects cells from damage. This is a simple image which shows the structure of cell wall. As you can see, cell wall, we can find plasmodesmata and middle lamella in the cell wall. Plasmodesmata connects the two neighboring cell walls. Plasmodesmata are the fine strands of cytoplasm which connect two neighboring cells through a fine pore in walls, while the middle lamella cements neighboring cell walls together. Now we are going to look at the function of cell wall. Cell wall provides mechanical strength and support for cells and the plants as well, and lignification even increases the strength of the cell. Cell wall also develops some waxy cuticle, which is used to reduce water loss and risk of infection towards the cell. Cell wall supports the plants, and especially herbaceous plants, through turgidity. They can prevent the cell from bursting due to osmotic pressure. The other function of cell wall is to protect the cell from physical damage, as well as they control the growth of the cell by limiting the cell size and shape. These are the quiz questions that we have prepared for you all, and these are the answers. Thank you.